For our very first session this morning, we're calling it the Frontiers. And George really talked about the technological frontiers of the PGP, and there are also geographical frontiers where we, as a research study, are growing. The Harvard PGP only accepts US citizens, and so we are organizing a federation around the world of open public genomic research studies, and we're very happy. We worked with a wonderful group in Toronto who's here today. We spent 2 years organizing the study in Canada, and it launched in December with a really amazing week-long series in the Toronto Globe and Mail that I really encourage you to look up. And we have the journalists who helped organize that here to interview Steve Scherer and Jill Davies. And so first, I'd like to welcome Steve Scherer on the stage, who's going to give a few minute overview of PGP Canada. Steve.
Well, thank you very much, Jason, and also George and the organizers for this wonderful meeting. It's great to come to Boston. What I'd like to do is take a few minutes to give you an overview of our Canadian Personal Genome Project, and then I'll also introduce the panelists that will be here for the next hour discussing some of the issues that are specific to our project that may be of interest to you. So by way of introduction, our moderator for this session is Carolyn Abraham. Carolyn is a journalist and author. She is a geneticist specialist, I would say. She probably knows more about genetics than many card-carrying geneticists. And what we've all identified to you is something interesting that we found in our genotypes. So Carolyn is a chromosome 11 inversion carrier that she'll probably tell you a little bit more about later. She's also the author of a newly released book called The Juggler's Children, which is shown on the left of this slide here. And I would encourage you to look at this. This is the story of her genetic ancestry with respect to answering long-standing questions in her family history. And I think, as George pointed out, and as we all know, family history is very important for medical history, understanding the environment and the contributions of medical attributes in our own personal medicine is perhaps the most important information that we have to pass on to our children until we fully understand our genome, that is. So secondly, we have Jill Davies. Jill is a genetic counselor by training, and she's head of genetic counseling at the MedCan clinic in Toronto. As you know, the vast majority of healthcare in Canada is a socialized healthcare system administered through the province, and I think this will come up, the provinces, this will come up over and over in our presentation. Who the payer is will influence how we actually roll out personalized medicine. But Jill kind of works in between because she's working in a private healthcare clinic, and her genotype she disclosed was she's an APOE4 Alzheimer's risk allele carrier, so perhaps she'll talk a little bit about that. And finally, my name is Steve Scherer, and I'm a genome scientist, PhD trained geneticist. I work at the Hospital for Sick Children in Toronto, and I direct the Genome Center there, the first and I think it's the largest genome center in Canada. And I also, relevant to this meeting, direct the McLaughlin Center, which is a $100 million initiative at the University of Toronto run through the medical school, and the focus is genomic medicine. And one of the primary focuses of our investments have to do with education, in particular in the MD-PhD program and in genetic counseling. So the impact of how this information will roll out in the healthcare system and in the public realm. And maybe we'll come to this a little bit later, but I have some very interesting chromosome 11 copy number variation deletions that I think make me somewhat unique in the population. So we've been working on putting together the Canadian version of the US PGP for actually, probably, I'm trying to remember how long it's been now, but certainly since 2007, maybe a little even before that. We tend to do things a little bit slower, perhaps more thoughtful. That's for you to determine. But we spent a lot of time, and it's also harder for us to raise money to do this kind of thing. But eventually it was launched, and Carolyn Abraham will tell you a little bit about the launching and why the Globe and Mail. The Globe and Mail is, I would say, the equivalent of the New York Times in Canada. It's dubs itself as Canada's national newspaper. It is the one paper that's printed and red coast to coast. So if you're on the cover of the Globe and Mail, it has probably the most impact. But we eventually launched things in December of last year formally. The logo for our Canadian project is shown on the left here, the DNA strand looping into the Canadian maple leaf, and then underneath are the A's, G's, and T's that form the outline of our great country of Canada. You can see Hudson Bay kind of in the middle there. And this was designed by one of my PhD students. So it's good to have these students who are very creative in presenting your data. The Globe took great interest in this, and Carolyn will talk about this. They ran a series of ads and articles for a whole week or so. Ads, and this type of ad here, it says, "You were always taught to share, now hand over your DNA." These were all over bus stops across the city, and of course, and run in the paper itself, and many other places, on the subways, etc. And I think from the Globe's perspective, they were trying to identify a series a story to dissect. And really the question they were trying to raise is, why is Canada the only G8 economic country that has no law or no legislation around genetic discrimination? Was it because, or is it because there's no need for it, because we're in a socialized healthcare system? Or we just haven't got to that point yet? And this was really the question that they were raising through this series. So the PGP Canada is a joint venture between the University of Toronto McLaughlin Centre, the Genome Centre that I direct, the Centre for Applied Genomics, Sick Kids Hospital, and MedCan Clinic, and many other partners, and also, very notably, the US PGP. We thank Jason and George for their support and efforts over the years, getting us the necessary information with respect to consents and the project design and things. We couldn't have done it without them. We just apologize it took us so long. So, why is the Canadian project different? So as a Canadianization of PGP-A, of course, we'll only enroll Canadian citizens. So there was certainly, I think it's a few hundred Canadians who were interested in the US project who were on a list, but they could not enroll. So this gave us some of the impetus to start a Canadian project. Since the Globe series has run, we've now identified over 700 new registrants who are going to participate in the project, and roughly, I think the number is 20% are going to be paying for their own genome and all of the cost around that. So we're prioritizing those. We intend to do 30 full genomes in year one. We wanted to do this very conservatively to roll things out in a way that we could manage expectations properly, get all the data, and do things in a very thorough and informed way. I think what's different with our project also is we provide genetic counseling to participants both before and after they get the data. Okay, so we'll come back to this a little bit later, and I think Jill will probably want to talk about this. We require the participants to discuss with relatives their enrollment in the project. So they actually have to designate in their consent that they've done this by checking a box. We don't enforce it in any way. We don't tell them who they have to talk to, but we do make it a priority for them to try to address. And I think that's a little bit different than here, although I think it's strongly suggested. I think we just try to put a little bit more of a stamp on it. We have a different website and different administration, but we want to work as closely as we can to share experiences with the US and other projects coming together. And I think importantly, and I hope this comes up in the discussion throughout the day, is we will have a different annotation pipeline. And when I talk about the term annotation, for many of us who are scientists, we think about the algorithms and the databases and that we use to compare and contrast to. But I think annotation goes a little bit further than that. We're talking now about using health records from a different healthcare system. Access to information is much different. The way Canadians deal with the information, share the information is much different, which impacts, of course, is based on the cultural differences between our countries. And I can tell you firsthand, the so-called incidentalome that George mentioned earlier, those variants that can be medically actionable, but that may be identified for incidental reasons. The way we look at those lists are quite a bit different because the way we actually act on these variants is different in our country. So I can tell you, maybe I'll get into this later, but my group studies the genetics of autism, and when we sit around on Monday mornings with a multidisciplinary team looking at the data, everybody sees something different in the data depending on what your background is, what information you have access to, and how you interpret that. So how we identify those so-called actionable variants is quite a bit different from site to site, depending on who's looking at the data. different experiences with the data. We use different pipelines. We have a lot of unpublished data, and hopefully, again, I'll come back to this. We're doing the annotation within our genome center setting that is within a medical health center. And we run actually 10,000 genome-wide experiments a year in our center. Mainly clinical microarrays, but we've moved now into exome sequencing and whole genome sequencing. We have a lot of unpublished data, a lot of experiences, and it's the same team who's going to do the PGP annotation. So we're bringing a lot to bear on these annotations. And as I'll show you in a slide, we also are not just looking at single nucleotide variants and small indels. We're looking at larger structural variants, which I think will have a great impact when we're trying to interpret the role of, in particular, rare variants in genetic disorders and personal health. And finally, the impact on the health management may be much different. As I mentioned, we have a different culture. It's a lot different in Canada, the way we deal with this type of information, and a different payer. So healthcare is administered through the provincial ministries of health. That's who I have to answer to when we're kind of dealing with this kind of thing. I had to show, I'm a scientist, I had to show one scientific slide. This is work that my PhD student, Andy Pang, has done with Craig Venter's group over the years to annotate, come to the question of how much genetic variation is there in a single genome, and this is Craig Venter's genome, which has been sequenced using Sanger sequencing, and now using multiple NGS platforms has really been sequenced to death. And Andy could kind of categorize the size of the variants as shown on the x-axis, and then the number of variants in each of these different size bins is on the y-axis. So the single nucleotide variants is the red bar. Each of us has roughly 3.2 million SNVs, I'll call them, SNPs, because a lot of these are rare variants. And then we use a lot of different technologies to try to annotate all of the other copy number variation or indel type data. And you can see that there's a relative equal distribution of these variants based on size across the genome. And the point I wanted to make, in fact, that there's 10 times as much unbalanced variation, so CNV or indel in the genome, than SNV variation. So if you're trying to annotate in a personal health perspective, you need to actually account for this variation. And in fact, even in complete genomics, the data is absolutely spectacular. It's the best data that we've been able to use so far. But even that source only actually accounts for about 50% of the indels and CNVs. So I think we need to get better on the annotation side. And I'll talk a little bit about that, I think, in my own genome. And then finally, the Globe ran this survey that's on the web, and I would encourage you to go look at this. There's been now well over 1,000 participants who have kind of looked at the questions that they posed, with the ultimate question, would you have your personal genome completed? And overwhelming answer across the demographic age groups is yes, they're very supportive, and they would make this information available. So I think we'll come back to this. Okay, so with that, I'd like to call Carolyn and Jill on the stage and turn the microphone over to Carolyn. So this is Jill Davies and Carolyn Abraham.
Thank you very much for that, Steve, and thank you very much for having us here. It's exciting to come out and talk about the Canadian experience with the Personal Genome Project. I wanted to start off by saying that I first met George Church actually at a sort of a town hall community discussion at the University of Toronto that was held, I don't know, 6 or 7 years ago, and there was discussion then about what the Personal Genome Project was, and how people in the United States were being asked to step forward and offer their DNA so that it could be sequenced and put online, and it sounded very American. And the idea was, wow, was this maybe the basis of a reality show, and just couldn't see something like this flying in Canada? We're private, we're cautious, aren't we? And yet just a few years later, Steve was already working in the scenes to bring this idea to Canada, and I remember going in and having an editorial meeting with Globe editors about this idea, and really the view was, will people really do this? Do you think enough people will take this leap into the unknown? Do they want to know this about themselves? And you have to understand that it's a bit different in Canada because we're the only G8 country without a law protecting genetic information. There's no GINA equivalent in Canada. So yes, we have a public health system, but still it brings to bear questions of employment and life insurance and other issues that obviously come to bear when you talk about your personal information in this light. But as Steve showed you in that last slide, the response was overwhelming. I think one of the interesting things in the way that the paper decided to approach it was and I'm going to be asking Steve and Jill about these motivations, was to see this as, yes, about you, the individual, but also about the contribution that people were being asked to make to push this science forward. So it was framed very much around the idea is, even if this can't help you in the here and now, genomic knowledge is a bit like that rushing stream, and if you step into it today, it might be a different, water that you're stepping into altogether, a week from now. So what implications might this have, your contribution for future generations, or maybe not even that far down the road, maybe just next week? And posing it as a question, I think, engaged people. And it generated, as you can see from some of the slides and some of the responses, a pretty dramatic response, I would say, in favor of moving ahead, which I think was surprising to some, certainly inside the organization, and to others as well, who thought, wow, the diversity of opinion was very well argued, and it really engaged people. And I think that you saw in fairly short order how many phone calls came into the project. Certainly, we got emails. And before I talked to Steve sort of about the genesis, and Jill about sort of going where no Canadian had gone before, because she is, as we called her in the Globe article, Canuck #1.
Forgot to mention. She's PGP1.
PGP1.
PGPC1.
Yeah. I recently wrapped up what I would call my own little personal genome project. Steve was kind enough to just show a slide of my recent book, but I bring it up here because there's a lot of talk always about, sort of the genome as crystal ball and looking into the future. And I was really keen to solve a couple of mysteries of my ancestry that had been with me for a very long time. And my mother only agreed to participate. It took 2 years to talk her into it in some ways. But if we used the genome to look back, to really look at it only as a portal to the past, and take the markers and the information that we saw from that to find answers that would solve mysteries of our heritage. Without going on too much about that, I'll just say this by way that, I wasn't adopted or anything like that. It was mostly that I'd grown up in a fairly, I'd say, white neighborhood, but, we were immigrants in the 70s to Canada, and I was, the sort of the brown girl with the Jewish last name who went to the Catholic school. And so it was a real mix. And then when I was about 10, I discovered that my grandfather looked Chinese. And so then I really wondered how we got a name like Abraham. And with DNA, I was able to, look at Y chromosome testing and other markers in the genome to actually go back and amazingly, sort of solve those mysteries and ferret out those stories. And one thing I found that people don't often think about is when it comes to DNA, and I'm sure many of you know this yourself, genomic information, when it comes to how it impacts your sense of self and your personal identity, really has the power you are willing to give it. How important it is, I think, in the sum total of who we are and the product of our culture and experiences can be informed, can be upended, but I think ultimately can help in terms of self-knowledge, and also offers this wonderful, I think, and often overlooked ability to connect us with strangers and remind us just how close we are as a species, how young the species is, and how remarkable it is that we are essentially all part of one family, and in some ways diving into the genome allows you to look back for that. So with that, I'd like to turn this over to Jill and to Steve to talk a little bit about the Canadian experience. And I'd like, first of all, to ask Jill. I mean, I talked about my own personal motivations looking at genetics, and I'm sure each of you out here had your own. But Jill, if you could tell us a little bit about why you decided to take the leap.
Mm-hmm. Well, I think Steve gave a pretty good introduction of my background, but I'm a genetic counselor working in a pretty unique setting. So it's a primary care setting, it's a preventive care setting, and it's also private. So there's a team of genetic counselors who provide counseling both pre and post-test for individuals who are going through personal genomics. So typically that's been SNP-based testing until recently, and will continue to be for the next little while. But given this experience, when Steve and I sort of started talking about working together on this project, we initially thought that the clients or the individuals who we meet with at MedCan at our clinic, and we've seen about 4,000 people to date, might be a good pool to draw from for this project. And so, so that was sort of the initial stages of the conversation. And then it was a couple of years going through REB approvals and really getting to where we got to last fall with everything being approved. And I remember the day, I remember where I was, when I got the email from Steve and from Michael, the ethicist on the project, saying, you know, we have the opportunity to provide you with a free genome. And Michael, who's always the kind of diplomatic ethicist, said, no pressure. He's gotten to know me better and knows that he wouldn't have to say that, and I obviously jumped at the chance. At that time, though, I didn't know that I was going to be the first in Canada, and I didn't know that the media was going to build such a big and excellent story around it and bring such media attention to it. But I think probably one of the most important pieces, and one of the things we talked about early on, was the importance of having genetic counseling involved in the whole project. And so making sure that everyone who goes through our version actually receives the education, the interpretation, as much as we can anyway, around this.
How do physicians, like, I mean, you've dealt with patients a long time, how do physicians, doctors, receive information that patients are coming to them with after having had sequencing done?
Yeah. So again, I'm pretty fortunate that I do work in a progressive clinic. I work with a team of about 60 physicians, and we really integrate genetics into that as much as possible with pretty open feedback from the physicians. They tend to rely on me, so often if the patient comes to them with questions, they'll divert it back to me, but there are a few that have taken an active approach in really educating themselves and learning, and I think the key area they see benefit is the pharmacogenomics. So there's a lot of utility around that in our population. But for the most part, when patients take it to their physicians outside of MedCan, they don't get very positive response. So
What did doctors say to them?
They send them back to us. So for the most part, I think they don't want to have to deal with the information, or they don't know how to deal with the information. So I think, and Steve will be able to speak to this further, but part of our project is really to try to overhaul the medical school curriculum and really get the education out there for physicians. And again, we have a few that sort of lead the way, but there's a lot more that needs to be done there.
And Steve, that brings me to my point, is why is it that you decided to do this? Why take this on? What were the maybe scientific reasons driving it?
Well, so the three major reasons were, one, it was clear that genome-wide testing, experimentation, was going to have a huge impact. I could see that from my research. And to really understand how that would impact the general population as a whole in Canada, we had to do the project in Canada, because to understand the Canadian questions, we had to do the Canadian version of the project, as I tried to point out in my introductory comments. And we've seen that over and over again, from the consenting to the interpretation to how people want to understand the data. I mean, you know, you can explain to them whatever you want, but their ears are only be open to certain aspects of it. So that was the first thing. We wanted to have our own Canadian experience. And the second was, you know, I've been working in this field for 20 years or so, and we've done genetic association studies piecemeal to date. And this is the so-called perfect genetic experiment, where we get a whole genome sequence so we can do things properly. So it became very clear that this is going to change medicine. And in fact, so much so that the head of our molecular diagnostic laboratory at our hospital, which is the largest one in Canada, because again, diagnostics is delivered mainly through the hospital-based system, was coming to me and to the Genome Center for answers more often. And now, as I pointed out earlier, I think there's roughly 10,000 genome-wide tests are done a year. And the interpretation is done by whoever can best look at the data. And the third reason was really this issue of control data, having population control data. It's held up so many of my studies in the past, and having a project that is going to publicly release the data so you can go yourself directly and get it downloaded from the web and also have access to health information. It will, once we get the numbers in place, revolutionize things. So right now what scientists, practice scientists do is we kind of cobble together control data. We go to the NCBI, those are the database that the world uses in the States here, and I get 10 emails a day from NCBI to report on the different control data sets I use for the different, it's killing me.
Wow.
So it's going to change things once we get in the tens and hundreds of thousands numbers, and that will come with time. I think the real challenge, of course, is that we've only got like 5, 10, but I think once we get the process in place and we use Scherer data, we'll get to those numbers and sequencing comes down. It's going to go fast.
Yeah, well, what do you think about the gap that obviously Jill's described and other people talk about? I mean, I was at a conference last week where bioethicists are very concerned about technology not only outpacing, the ability of, policymakers to deal with all of the kinds of information that come out of genetics, but that science has been, sort of overshadowed by technology pushing us in this direction. And what should the medical systems be doing? What should doctors be doing? Mm-hmm. How would you sort of describe this in terms of the challenge and what PGP has to offer?
Well, again, I think a lot of the data that's going to come that we need to present to the policymakers will come from this type of project, as it typically does. It's the initial data that piques people's interest, and then these types of things have to go through health technology assessment studies, which are long-term. But traditionally in genetics, in medical genetics, once a technology comes about that gives you a better result, a more informative result, a higher resolution test, it becomes a standard of care just because the demand for that test is there. So again, this was to force those questions. We introduced it so we knew genome sequencing was kind of an endpoint. It shouldn't get any better than that. I mean, it's not perfect as I showed you, but it's getting there. And then we want to work backwards. So we know we have the data, the technology, and let's figure out all the questions around it.
Right, right.
People, the politicians, they like to talk to scientists. That's what I found too. Scientists, even if they don't know the right answer, they want to get they understand that the science is the truth. Right. So they want to know what the truth is, and then they'll try to make things work.
Right. And Jill, maybe you could talk a little bit about your personal experience with looking into your own genome and find the early findings so far. I realize it's still undergoing analysis.
Yeah, we don't have. I mean, Steve has a lot of information back. I don't think we've really sat down and gone over it, but I can speak to my kind of experience going into the project, which is, of course, I consent people to do genetic testing every day. I go over consent forms with them, but I think with me going into this project, I hadn't really read the consent forms or gone through them until I do everything at the last minute until we were just about to draw the blood sample. And I definitely had a moment in time there where I wasn't sure if I was ready to jump forward, but then of course I thought, yeah, I absolutely would. And I think we had spoken about the issues around even my family understanding this information and what it means for them. And it was a learning experience for me because I counsel people all the time in the clinic about, this is information that's relevant to your family and you should share this with them. But I don't think I was quite prepared for my family's reaction to this information as well.
In what way?
Well, I think they were far more concerned than I was about the issues with privacy and non-discrimination. And of course, I just blamed the media for overhyping those issues and said not to worry about it. But in terms of the data, I think really we've surprised me, only found one significant variant that's a recessive gene. I've had a lot of, prior to doing the whole genome sequencing, I've had a lot of other tests. I've had pharmacogenomics, navigenics, pathway 23andMe, I've had them all. And I think the biggest finding is what Steve's already mentioned, which was the ApoE finding. No family history. So it's one of those things that I think about, I live with, but
has it impacted you in any way?
Yeah, I think, again, I counsel patients kind of daily on outcomes of these kinds of tests and what they can do on the preventive side, on the proactive side. And so certainly it's, it's something that I'm aware of in terms of early detection or potential for early detection. I don't have to worry about it, luckily, for another potentially 30 or 40 years, but it's something that's on my mind.
Right.
Yeah.
But knowing this, I mean, I guess what I'm asking you is, does it change the way you feel about knowing the information? Are you glad you do know it?
No, absolutely. And I mean, I knew that before I went into whole genome sequencing, where I had potential to learn bigger and potentially more significant issues. And no question, I would do it.
Okay. And Steve, you've also done yours.
Okay, but maybe can I just preface one thing?
So
we got really rushed because the Globe Mail set this aggressive deadline, and when they wanted to release the articles, we didn't expect we'd have to get Jill's PGP C1 genome done so fast. So in fact, we've done a lot more annotation now, and this is one of the things. We built a new browser to actually present the data to clinical geneticists and counselors and individuals. So we'll go over that with you. And I've peeked at the data this week, and there's some more interesting findings there. But this is going to be one of the challenges. This is really the challenge is the re-annotation, the constant re-annotation of the sequence. The sequence will be essentially static. It's going to be the re-annotation. That's the big challenge, actually. We all know that, and there's businesses developing around that. But to come back to my genome.
Yes, you.
Yeah, so...
more about you.
So, my genome is kind of famous in the laboratory. It's, a lot of people didn't know it, but I'm 999-1. I love that slide of it. It's, share your DNA. When I was a graduate student, when I first started in 1987, the first day I landed in the lab I was doing a rotation is they said, okay, here's your lab bench, now come with me. And they sat me down and said, you got to give us 100 milliliters of blood because we need DNA to make libraries from. These are the old days. We used to make phage and bacterial libraries and things. No consent, nothing. So that DNA sample from over 20 years ago, it's still kicking around, and we use it as a control for everything. So it's been sequenced to death. Most people didn't know it was my genome.
Wow.
Okay, so then, of course, we had the genome done, and it's been mainly annotated with respect to SNVs and SNPs. And I have a very, very boring genome. Okay, it's exactly what my parents told me. I was kind of hoping more to have a Heinz 57 genome like you. But it's very, what we expected. But then I asked the guys who know a lot about doing annotation for these copy number variants to look at it. And I didn't tell them what it was. Just throw it in with a batch of other samples we're doing. For those you don't know, my laboratory's sequencing, we're doing, I think it's roughly 1,000 whole genome sequences from families, 1,000 families right now in our autism project, so we just kind of throw it in there. And the annotation comes out, and I said, can you look at this batch of samples? And Christian, who I think both of you know, comes back and he said, man, this is one sample. It's like really messed up as my genome. So he looked at so on chromosome 11, I've got these two large deletions that are nearly side by side, and one deletes one copy of this SUCL-A2 gene. If you have two copies missing, or if it's recessive, it's a mitochondrial DNA depletion syndrome. Like I wouldn't be here right now. So kind of humbling to think that, I've got one copy keeping me balanced right now. And then the other one, the other deletion is like a 100,000 base pair deletion that knocks out this gene no one knows anything about, other than in Drosophila, it looks to be involved in brain development. So I kind of keep my eye on that one to see if there's any new papers that come out. And there's lots of other stuff, but those were kind of unique because I have 3 brothers, and we've looked at our whole family. And I inherited that one, chromosome 11, from my mom, so she thinks this is very special. Like this is what we share together, these 2 mutant alleles. So, but anyways, you know, it's not something I dwell on, but I'm very interested in, and this is what we do, is to develop the tools to re-annotate the sequence and see how the interpretation evolves over time. And I'm convinced at some point they're going to have something going to change my management of my health.
And on those major deletions and those copy number variations, I was really fascinated by how much these can differ by population. Can you talk a little bit about that? Like the copy number variations were things you can see in one group and not another group, and the importance of interpretation. Like when you look at something that looks clinically significant, maybe in one group, and see that this might actually have, or believe this might have a deleterious health effect because it's large or it affects a very important gene, but yet if you look at a subset of another population, find that it is, in fact, common.
Well, I think any genetic variant is like that, but it's just that the impact and the impact on disease and traits, and which of course will then be open to different selective pressures, is greater than a smaller variant because the whole exon or whole gene or maybe present in one copy or 3 copies or something like that. So it's just a bigger lesion that nature and evolution have to deal with. So that's what I try to point out on my slide. You have to really annotate these things. I think, you know, what's come out of the medical genetics field in the last few years, which really hasn't got out in the public media yet, is that many common diseases are collections of rare disorders. And that's certainly the case for neuropsychiatric disease, mental health disorders, things like that. And we're seeing it more and more as we look in some of the chronic disorders that afflict us. So that's the power of genome sequencing, is you can directly assay these. You don't have to try to impute them by looking at how they reside on the chromosome by a common allele or something like that. You directly assay them. So I think that's going to be the real impact, is assaying these rare variants and then doing the disease association studies. What we're seeing coming out of the exome sequencing projects that are going on in the States and around the world is almost whenever a disease-causing allele is found or mutation is found, it's in a gene that's already associated with another disease. It's just that the whole clinical spectrum of that disease gene has not been cataloged properly yet. So I think as we do the sequencing, we're going to redefine how we categorize diseases, maybe regroup them, reshape them, but we're going to find more associations, and that's what I think is really the most important thing.
So that a person might have their own version of X or whatever.
Yeah. But to find those rare variants, you got to find them all. You can't just assume that they're all going to be in the SNPs, because that's not the case.
Right.
The missing heritability, a lot of it's being found in these other variants.
Okay. And Joe, what's your final sort of perspective on how the patients that you counsel deal with sort of this, you know, moving field and the ambiguities? I mean, what's your sense of their perspective?
Yeah. I think what we've been offering them to date is pretty consumer friendly. So it's the kind of traditional SNP-based testing with interpretation and advice around prevention. I think when we get into bigger data sets like this, it will become more complex. And I think that's why we really felt strongly that we would want to have interpretation, genetic counseling around the results. I mean, genetics has always been an area where we've never been able to say for sure. I mean, even when we know we've said someone doesn't carry a gene, we can't absolutely exclude a risk for that disease. And so it's something that is just really needs to be built into the whole process of education and counseling individuals through this. So I think that we are planning to recruit a lot from our patients who have already had the first phase of testing. So they're already educated, motivated, have a specific interest in this area. So I think it's just helping them understand that this is the next step, and we're a long way from really figuring it all out.
Right. Well, I'm going to suggest, unless Steve, you would like to talk a little bit about where it's headed and how many people you think you'll have involved by.
Right. So I mentioned as a result of the Globe series, we had, I don't remember the exact numbers, but it was 600 or 700 people who directly contacted us. And what we've done is we've set up a committee to prioritize those individuals, because we can only process so many people through the genetic counseling, the ethics, and the examinations. The sequencing is not our bottleneck right now, although it will be, I think, at some point. Hopefully the costs come down so low that it won't be an issue. But anyways, we've got enough lined up who are willing to pay for their own genomes. And in some cases, we've got people who are going to pay for their own plus one more. So we've tried to recruit an underrepresented individual from our list. We've strategically selected some publicly visible people who are willing to contribute through education, public education, that kind of thing. So our first 30 are not going to be a problem. I think we'll meet that goal. And then through this committee, we'll prioritize the other ones. But funding should not be an issue for us for the next few years. I think we'll be okay. I think we'll probably need more human resource infrastructure to do the counseling and the ethics of consenting and things like that.
Right, right. And one thing I would just add to that is that there is a private members bill in Canada, no saying what will happen to it, but an effort to enact some kind of protective legislation around the uses of genetic information. But I guess uncertain, very early days. There had been an earlier one come forward that didn't make it before. So in a way, there's a sense in Canada that, because we have a public health system, we don't need to protect our genetic information because you're not ever going to be denied medical care based on something that your genome says. However, it's in the other spheres where that's a little murkier.
But there was no opposition at the political level by any of the three parties. It was just an issue of we probably don't need it. Why would we?
But we don't need it. Some people.
But I think this project actually brought it, resurfaced, and it's been tabled again this week. So I think there's a lot of attention in Ottawa, but it's because of this project and the recent investment Genome Canada made in personalized medicine.
Right.
So it's back there, and I suspect it will have some legs this time, but we'll see.
Right, quickly. Well, on that note, I think we'll open it up to some questions, if there are any, about the Canadian experience so far.
Are there any Canadians in the audience?
I guess we'll ask for people to come to the back for questions. And I'll take advantage of this opportunity. Sean Bradley from Brandeis University. I hear a lot about an emphasis away from SNPs or SNVs, and I wonder if there's a magic number as far as sequencing length in order to find CNVs and other variation that you're referring to.
Yes. So there's a lot of workshops that go on around this question, and the X Prize actually had a few workshops a few years back to talk about this. And in the field, it would be, based on my experience, it would be great if we get 10 kb or greater reads. It would really kind of probably address this issue of the resolution gap. Sanger sequencing reads are pretty good, so if you can get in the 1 kb range, I think it would resolve a lot of it. But the longer, the better. There are some technologies, I won't mention any because I always get accused of showing favoritism. But that will give you longer reads, and that's something that you would like to do. I should say that for all of our genomes, we're also running microarrays, so we do that in-house, which helps us check accuracy of sequence, and also it helps us in the CNV calling. And we're not doing karyotyping though right now, although in some cases that data exists. But yeah, the longer the read, the better. I think with respect to coverage, we're typically going in the 40x coverage range.
Thank
you. Wondering, will there be any attempt within PGP Canada to track the impact of the personal genomic sequencing in terms of follow-on healthcare? So tests that might result from findings of uncertain significance.
Yeah, so we actually have a project ongoing at MedCan right now that's been generously funded by Steve's McLaughlin Center, where we're looking at our internal data in the same way. So what are people doing with the navigenics pathway, the SNP information in terms of behavior, outcomes, and then what's happening downstream in the healthcare system. So it would be nice to follow that same kind of project in this area, but I don't think we have a specific plan in place to do that yet. It'll be interesting. We're still in the process of collecting our data. So we've put 4,000 people through our test over 4 years, and it's a 2-year project. So I think it'll end. All the data collection's just been complete, so we're doing the analysis on it right now. So I think that will help to inform what we might do on this end as well.
That's a really important question. I'll just flip it a little bit. In our clinical research studies we do in genetic diseases, the question I'm asked from the hospital is the same, but it's coming from a different direction in that. So you're finding these disease-causing genes, and we can facilitate a diagnosis. And now you've added on the fact that you're getting not only diagnostic tests for the clinical question at hand, but also all these other incidental findings. And in fact, I think probably many of you know, 50% or more of the clinically relevant findings will be questions that are not targeted to the disease you're actually pursuing. So when we sequence a genome from an autistic child, more than 50% of the time we find something else that has to be looked at. So what the hospital wants to know and what they're doing their impact study on is how that impacts the downstream management of that child or that individual, and what clinics are impacted and how they have to prepare for this type of thing. So that's the real issue. Does it impact imaging? Do you have to go to neurology? Do you have to go to these different clinics? And that's where the real impact is. And I think you all know, but just to reemphasize that when you do the genome, you're not just getting the data on one gene, you're getting the data on 23,000 genes.
Right. I think there is a study going on like that with whole genome sequencing in kids in Canada and finding out generally sort of what happens, but it's very early days.
Right.
Right. Is that it? I think so.
Well, why don't you tell us? Tell us more about your book. This is I think we have still a few minutes, right? And so Carolyn's very humble. I think the story is great. I'd like to hear a little bit more, because when we started this, what I didn't mention is it was a lot to do with how we deal with the data, the medical perspective, but it was an experiment, a social experiment too. And I think that's really your story.
Well, I mean, I'd like to see. Thank you, but I mean, have many of you out here done ancestry testing with your DNA or looked at ancestry questions? And I guess I'd gone to it with very specific questions, wanting to know if it could actually, if I could actually find out whether or not my great-grandfather or my paternal heritage was Chinese, and whether or not it could lead me back to a name. And because my great-grandfather had been this figure in my past growing up. He was a circus juggler who appeared in South India in the 19th century and then disappeared, or that was the story. And it led me, obviously down many paths on one side of my family, but one thing I found very interesting about this process was certainly with people who do a lot of genealogy work or genetic genealogy work, was how much they are pushing the field forward themselves. Doing these huge surname projects and collecting Y chromosomes and, being able to calibrate their own sort of molecular clocks within their families and know, sort of how and when mutation rates are working and identify what they were calling naughty alleles. And in a sense, working on these big projects and coming together and sharing this information with each other, and yet on the other hand, so separate and far outside sort of the traditional scientific realm. I found it especially interesting in terms of bringing this information out in my own family, where you run into ethical issues as a result of it. I'm sure many of you know this, but like in terms of issues of false paternity, and you think you're doing this, and this is an effort to answer your questions, and yet it has an impact on everyone who is related to you, and people you didn't know were related to you until this kind of testing shakes them out of the family tree. And I find that, with DNA, it's just this incredible molecule of information that tells you about your past and your present and also your future. And interestingly, you'd think that having covered this for as long as I had, that I might have signed up for PGP in Canada, but I actually have not, in part because of that promise I'd actually made to my mom about I would do this, that her participation rested on that condition, that we'd look back and sort of not forward for now. But anyway, if there's anything else or no more questions, and
Do you have anything else? Good.
okay.
All right.
Thank you very much.